Hey, off phone. Just in case. Right, guys. Happy. Me, thank you for making me, happy because I was really sad. I hope that you might not know that I had a huge accident. My vehicle is under the repair now. Our lady කෙනෙක් ඇවිල්ලා බයික් එක නෙමෙයි මේ PS එක පිටිපස්සෙන් ඇවිල්ලා සාමාන්‍යයෙන් පැයට කිලෝමීටර් මං ස්ලෝ කරලා නතර කරගෙන වගේ යනකොට පැයට කිලෝමීටර් 60ක 50ක වගේ ඇවිල්ලා පිටිපස්සෙන් ඇප්පුවා. අප්පුපාර මම ගිහිල්ලා ඊට ඉස්සරහා තිබ්බ කාර් එකට ක්‍රෑෂ් වුණා. වාහනේ ඉස්සර පිටිපස්සත් ඉවරයි ඉස්සරහත් ඉවරයි. දැන් ඒක රිපෙයාර් කරන්න දාදින මං මෙහෙම ඉන්දියා නෑ මං ඇතුළේ ඉඳ පින්ද මට මොකුත් නෑ. වාහනේට තියෙන තුවාලයි. මට තුවාල දාන්නට අමාරුයි වාහනේට තුවාල දාන්න. මට තුවාලෙන් අවුලන්නේ. හා මේ ඉන්දියාවේ සෑහෙන මේ අභ්‍යන්තරින් තුවාල වෙලා ඉන්නේ. ඈ එමුනේ. And thank you for making me happy even for නේද අර ඒච්චර sad එක නැති වෙන්න මේ ඒ දීපු happiness එකට thanks so much and this will be remembered forever, and the major thing I have to say is you don't have to make me happy with the gifts, because you are the biggest gift that can ever be, right? Always my friends are the biggest gift, so and I'm really happy with your people and that is completely more than enough. And other thing is if you want to repay me something or you want to treat me something, that is improving yourself with the knowledge, with the subject and having good grades and someday you get an A result, you coming to my class and you say, sir, mama ekat thina, I got an A, now I'm going to university and I'm in this place now because the things that we learnt in the class I have improved a lot. This level, I'm now in this level. If you say someday coming to this class, then that will be the biggest treat that I can ever receive, and I'll be doing my best to make you to that level, right? I'll be giving my best to take you up to that level, and hopefully you're on the track, right? And in the future I hope that you will be doing really good, right? And in the future I might will be නේද සමහර වෙලාවට I'll be මීට වඩා සැර වෙන්න පුළුවන් sometimes, හරි, and මීට වඩා හොඳ වෙන්න පුළුවන්, මීට වඩා විහිළු වෙන්නත් පුළුවන්, හරිද, මීට වඩා විහිළු වෙන්නත් පුළුවන්, depends, right? Depends, but all what I do is only because that I want to see something better in you, and I want to see a better future in you, that I'm seeing a future in you that you might not see it yet, right? Because I have seen much more things than you, so I'll be doing things to make you a better person. That's the only thing that's in my mind and in my heart, right? So I'll be sometimes angry, I'll be sometimes nice, I'll be sometimes gentle, I'll be sometimes tough. That is only because I want to see something better in your life, right, in the future, and I'm seeing it and I'll be doing it as well, right? I'll be giving my best efforts. So bear with me for 2 more years to come and try to do the best that you can do and make everyone proud, right? And thank you for the cake. अब मैं कट करूँ, कट कर ला, क्या लखाऊँ? Oh, एक डिक्लोफेनाक क्या किलो हम बस डिक्लोफेनाक ना एक को इट बस मांगता माता को मांगी मैं भी कहती हूँ डिक्लोफेनाक क्या? End up me come. In. Kau lagi di chicken ini kawan. Thank you for the cake. Thank you. I can't know. खाना हो गया Thank you, Pati. Thank you. Thank you. Thank you. How it going? Thank you for great.
Have this.
Thank you. They are rising in Kevin. You two girls come. You come in. Come. You come. People don't see. Only 3 or 4 people are there. Come. These are the 2 girls in our class. This is yours, huh? Thank you. And this is the other girl in the class. Thank you. That was Diana. There will be a lot of messages on this. Right? Yeah. So Atifa Singh's are lady in Sally Gatta the Gileas. Atifa are lady in Sally Gatta the Gilego repair. They are doing repair because it's totally their fault. In front of the cool plant, you saw it, right? You're on the way. Yeah. So everything is there in my CCTV footage. So I was like slowing down. She came in all of a sudden like 60 kilometers per hour and hit me in the back. Huh? They had a damage as well, but it was like, huh? We know අනිවාර්යෙන් ඉතින්. Insurance එක මං කියන්නම් පස්සේ. මේ කපු එන්න. එන්නක මේ කප. එන්න කප මේ කපන්න. නේ. මේ කපන්න. එන්න කපන්න. Thank you, brother. Hasan. Right, shall we start? What was the last question? Make කරන්නේ. Right. So now, can we do a quick recap on what we discussed the last week? So now try to remember. Huh? What's wrong with this guy? Right. Listen, so a quick recap about what we discussed from the last week. We first discussed who are the microorganisms, right? And the size of them. And then about the omnipresence of them. And then about the nutritional types. Then the respiratory types, and we are continuing from there, right? So now in the omnipresence, how many factors were there? 5 factors were there, right? You have to remember all the 5 factors with the details. Bro, taking the whole thing in. Yeah. So and what else? So in the nutritional types, under the nutritional types we discussed how many nutritional types are there. So don't hit the camera. Ah, nice. Now tell someone to come to fix the camera. Yeah. That's okay. You tell someone to come. Right. So under the nutritional types we discussed 4 nutritional types were there, right? What were the 4 nutritional types? Photoautotrophic, chemotrophic, photoheterotrophic. Good. And you have to know the examples as well, right? I'm not going to remind you the examples, but you're supposed to remember all the examples, right? And when it comes to the respiration, we discuss there are 4 respiratory types, right? Depending on the connection with oxygen, right? Depending on the connection with oxygen, there are 4 respiratory types. Under that, we discuss the aerobic, then the facultative anaerobics, and microaerophilics, right? And the obligate anaerobic are the 4 types, and you have to know the examples of the bacteria and the chemical formula as well. Like when it comes to the aerobic respiration, you have to know the chemical formula, glucose breaking down into carbon dioxide and water, right, with reaction with the oxygen, reacting with the oxygen. And when it comes to the facultative anaerobics, they have like 2 respiratory types. One is when in the presence of oxygen, they respirate using the oxygen. That is the normal respiration, breaking the glucose into carbon dioxide plus water. And in the absence of the oxygen, the facultative anaerobics respirate through the anaerobic respiration, right? And that is converting glucose into ethanol plus carbon dioxide, right? So you have to study it, study and remember those. When I'm asking, you have to have the memory to tell. Memory එක තියෙන්න ටක් ගාලා හොම් සින්කින් ගාලා කියන්න පුළුවන්. You can tell now. Right? ඒ ගන්න තියෙන්න ඕන. So and we discussed that the anaerobic respiration of the facultative anaerobic is the fermentation process, right? That is the most important thing. That is the fermentation, and it's done by the Saccharomyces cerevisiae, the yeast, yeah, majorly, and there are some other microorganisms who can do that as well. And one of the microorganisms given as an example is E. coli. E. coli can do the same, but E. coli are not used for the fermentation like process, not in the industry, because E. coli is one of the bacteria who lives in the feces basically. Yeah, they live in the feces, they live in the dirt. Yeah, So you don't use E. coli, because there will be some other byproducts when you use the E. coli. Start drinking this as well. Right. Right. So what we have to discuss next is obligate anaerobic microorganisms. Right. Come to this part. Right. Obligate anaerobics. Right. So for the obligate anaerobics, they can't live under any of the oxygen environments. Like if there's oxygen, they be dying. They be dying due to the toxicity of the oxygen for them. So basically, the obligate anaerobics can only live under the absence of oxygen doing anaerobic respiration, right? Even the microaerophilics are the same, but the microaerophilics can survive under a certain pressure of oxygen. They can survive under a certain pressure, but above that pressure of oxygen, the microorganisms will die. And in some cases, it says that in a certain concentration, up to a certain concentration, they can tolerate. You know, the microaerophilic bacteria can tolerate up to a certain concentration of oxygen or up to a certain pressure of oxygen. Both are same. You will understand it when it comes to the rate of reaction. We discussed the pressure and the pressure of the reactants increase the rate of reaction if it is a gas. Yes. So because oxygen වල pressure එකයි concentration එකයි කියන එක තමයි අපි බලන්නේ. In a certain pressure, in a certain concentration of oxygen, they can live, but above that they will all die. That is microaerophili. The obligate anaerobic cannot tolerate oxygen at all. No? They can't tolerate oxygen at all. So they will get poisoned when the oxygen is present. Right? That is the basic explanation. Right? These two guys. We are late. Watch the recording. Don't hit the camera. So don't hit the camera. No cakes? Take off your cap. It's not the. Water? Right. So listen, obligate anaerobic microorganisms. The microorganisms only respirate under which conditions? Anaerobic conditions. They have to have anaerobic conditions. They can't live under any like in the presence of oxygen, right? They can't tolerate it. And the example, the only example given is Clostridium. You have to remember that. That is the example given. More examples were there, but it's not necessary because only the Clostridium is coming. So remember the Clostridium, that's completely enough. You go through all the past paper MCQs, only Clostridium is there for the anaerobic respiration, right? Nothing else is there. So remember this, that is enough, completely enough. And the example is what? Clostridium. And the Clostridium is used for the biogas production. We will be discussing in the latter part of the lesson, right? Right. So the other one, the microaerophilic, right? The microaerophilic. These organisms can only live under low oxygen concentration or pressure. When the oxygen concentration increase, it becomes poison for them, right? They can live under low oxygen concentration. This is important, or pressure, right? Or pressure. Can live under low oxygen concentration or pressure, and above that concentration or pressure, that microorganism will die, right? That those are called the microaerophilic. And like those 4 types of bacteria, like those 4 respiratory types of bacteria are very important, you know, because those bacteria have separate benefits for the humans. You know, when it comes to the aerobic respiration, the bacteria and all the other funguses, they are all doing the aerobic respiration. So now tell me a benefit of aerobic respiration that you think of. What is the benefit of aerobic respiration? So simply the aerobic respiration, what they do is they convert glucose into carbon dioxide plus water. So what would be the benefit of that? Huh? Energy? No, they don't produce any of the energy. They are not the producers. What is the use of it? Huh? What is the use of the aerobic bacteria and the aerobic microorganisms, like the any of the microorganisms that do the aerobic respiration? What would be their benefit? What do you think their benefit is? What do you think the benefit of the aerobic bacteria or any of the microorganisms do aerobic respiration? Basically, it's decomposing the garbage, isn't it? They are decomposing the garbage. The glucose together makes a carbohydrate, could be a huge part of a garbage in the environment. Ne? මොනා හරි plant එකක තියෙන carbohydrate වෙන්න පුළුවන්. Animal dead body එකක තියෙන carbohydrate වෙන්න පුළුවන්. ඔයා ගෙදරින් එළියට දාන මොනා හරි තියෙන liter වල තියෙන මොනා හරි garbage වෙන්න පුළුවන්. තියෙන carbohydrates වෙන්න පුළුවන්. ඒගොල්ලෝ මොකද කරන්නේ? They break it down into simple particles, and that's they keeping the surrounding very clean. If they were there, if they weren't there, the surrounding will be flooded with so many of the garbage, isn't it? So they are decomposing it. They are decomposing it means like simply they are cleaning it, isn't it? The microorganisms who do the aerobic respiration actually are cleaning the complete environment, and two of the main cleaners, the microorganisms in the environment are, one is bacteria, the other one is the fungi, right? Bacteria and fungi are the cleaners in the environment, right? So what's happening? Huh? Right? And what do you think the importance of the second one, the facultative anaerobics? What is the importance of the facultative anaerobics? Fermentation, right? Simply the beverages. People are having parties and everything because of the yeast, isn't it? If it wasn't for the yeast, so no party there. Nah? Huh? What? Cake? Even they take for the cake production? Yeast. Yeah. That is. Yeah. The bakery production, you mean? Yeah. The bakery production. Yeah. The most important part is the second one, right? Producing the alcohol, the ethyl alcohol. Yeah. Huh? Juice. Yeah. The main thing was like the first was the toddy production, the production of toddy. So if it wasn't for the yeast to produce the toddy or the ethanol, there will be no any of the there are uncle's parties, no parties will be there. So ethyl production, now that's a beverage, simply it's a beverage. So alcoholic beverages are produced because of this facultative anaerobic respiration, right? And the bakery products, actually the yeast are making the bakery products. So the bakery production, they help the bakery production as well, the facultative anaerobics. And third, we discussed the obligate anaerobics. So obligate anaerobics, what do? It was there as an example as well within the break. They are producing the biogas, right? The obligate anaerobics produce the biogas, which is methane, CH4. That is really useful for the fuels, like the biofuels. We make energy out of it, right? To sometimes to turn the turbines as well in some cases. And when it comes to this microaerophilic, like what do you think? Yes, the microaerophilic are the lactobacilluses basically. Lactobacillus are important for the dairy productions, right? For the dairy production. Yeah. So the yogurt, cheese, everything are made out of these lactobacillus bacteria, right? Yeah. Huh? Butter is no. Butter is not made of any of the microorganisms. Butter is made of the fat in the milk. You know, they take out the fat in the milk. They centrifuge it. Right, they centrifuge it and produce that short guy. Right, they centrifuge it and take out the fat and they do the churching and everything, mixing with the salt. They produce the butter. Right, butter production was there, but it's like not like it's out of syllabus now. Right. So come to this part. Did you highlight all this? Right. So come to the next topic, the growth and reproduction of microorganisms, right? The growth and reproduction of microorganisms, right? Come to this part. So we discussed this. The growth and the reproduction of microorganisms are like really rapid, you know, it happens really fast. When it comes to the growth happens really fast. So they come to a young age and they start to reproduce quickly as well. So now we discussed this under the omnipresence. Now what is the major reason for this the fast growth rate and the reproduction rate? Yeah, because they are small in size and their AV ratio is really high. They have a higher surface area comparing to the volume. Because of that, they have a higher absorbency and they grow really fast. Because they grow really fast, they reproduce really fast as well. So now this is why as we discussed, Lifebuoy says what? Only 99%, 9 can be destroyed. So the 0.01 will be left and that 0.01 will, you know, return the same size of the colony back again, maybe within 24 hours. Why? They have a very small time of like the generation time, or the time that it takes to double the colony is very low. Let's say like 1000 or like let's say 1 lakh of microorganisms were there. So of these microorganisms, if they are E. coli, within 0.35 hours, which means how much? That's 20 minutes maybe, yeah, within 20 minutes that growth will be 2 lakhs, right? And within 3 minutes that growth will be, like within another 20 minutes that will be 4 lakhs, and within another 20 minutes that 4 lakhs will be 8 lakhs. Likewise it will be doubled within very low, within a very short period of time. So every 20 seconds it doubles its size, right? So now let's think like they were like 100,000. So like in a 20 minutes it will be 200,000, like 400,000, 800,000, 16,000, 100 times. Like it is improving really fast. So within maybe 24 hours, though the Lifebuoy destroys the 99% 9 ne huh? Oh වැඩන්නේ දවසම ගන්න වෙනවා, daily ගන්න වෙනවා, ගගාම ඉන්න වෙනවා, ඉවර වෙන්න බෑ ඈ. ඔව් නාල විනාඩි 20ක් ගිහිල්ලා ආයි නාන්න වෙනවා, ඉවරක් වෙන්නේ නැහැ. හරි. That what is that simply says is they cannot be, you know, destroyed. That is the omnipresence, you know, they will be finding everywhere no matter what, they will be surviving ne අපි වගේ නෙමෙයි, they can survive, they will be surviving no matter how much we kill, they will be coming back, right? Yeah, that is how the bacteria, yes, තියෙනවා 100%ක් නැති කරන්න පුළුවන් මේ වලින්, radiation වලින් 100% නැති කරන්න පුළුවන්. But there's no point, because if it was exposed, if it was exposed to the if it was exposed to the outer environment, it will start to grow back in a very simple period of time. That happens, right? So you can't completely finish it. That is simply the omnipresence, right? They are found in everywhere because of their fast growth rate, huh? ඒ කියන්නේ සබන් ගෑවේ නැත්නම් ඉතින් රේට් එක වැඩි වෙලා අපි ඉවර වෙලා යනවා. සබන් ගන්න ඕනේ කියන එක නෙමෙයි, but you have to be clean නේ, you have to use something අද now the boys are dirt and lazy. Right? Sanitizer kills, no, they can't even kill the 100%. Only the radiation can kill the even not ultraviolet. You have to use something much more stronger than ultraviolet. Even ultraviolet can't kill the 100%. You have to use alpha radiation or something. Right? So come to the growth rates, the generating time. Shh, listen. Yeah. Is it important? What's the question? So life boy වලින් 99ක් යනවා. ඊටු 101 ආයේ හැදුවා නම් හරිනේ. ආයි හේදුවා නම් හරිනේ. It doesn't happen that way. ඒ කියන්නේ හරිද? life boy වලින් 99% අයින් වෙනවා. ඒක තමයි දැන් මම කිව්වේ. ඒ කියන්නේ life boy ගෑවන් පස්සේ 99% අයින් වෙනවා. but ආයි ගෑවේ කියලා ඒ ඉන්න ටික අයින් වෙනවා තමයි, but you can't do it at like there's a limit that you can expose to a that's like the soap is a see, so the girls know, yeah, because ඕන එකයි කියන්නේ දැන් basically the soap are the base, they are bases, so up to a certain limit it'll be like harmful for the skin. Your skin will start to makeup fall down වෙන්න ගන්න ටික ටික නේ. So you can't use it up to a certain limit that your skin will start to get damaged. Yeah. So like now this is the reason why they say we can only remove 99% 9. They don't say 100%. Why? They can't. You know, they can't. Because though they don't even remove 99%, they can't actually calculate that up to that level. So though they removed it, right, it will start to grow back within a short period of time. Why? They have a very small generating time. Within a very short period of time, they will reproduce the same number back within 24 hours maybe. Yeah. So what is the generating time then? Since microorganisms have greater growth rate, their generation time when it comes to reproduction is very low, right? Their generating time is very low. The table given below shows the generating time of few microorganisms. So what are the microorganisms? Now E. coli has a generating time of 0.35 hours, that is maybe 20 minutes or something, right? Now this generating time means the time that it takes to double the colony. අපි හිතමු 1000 ඉඳලා 2000 වෙන්න යන time එක double the colony. හරි, very short time. And the Bacillus subtilis takes 0.43 hours. Clostridium botulinum takes 0.58 hours, and the Saccharomyces cerevisiae takes only 2 hours, right? So usually they take a very short period of time to grow back their complete colony to return their initial size. You know, though you destroy the whole thing, within few hours they will return back to their original size. You know, that happens, right? Right. So that is one of the reasons for the omnipresence as well, right? Right. Next one, biological nitrogen fixation of microorganisms. We will be discussing this back again in the nitrogen cycle as well. Right? මේක nitrogen cycle එකෙත් discuss කරනවා. So nitrogen fixation කියන්නේ මොකක්ද? What is nitrogen fixation? Is nitrogen important? Yeah, why? For the plant growth. It's like the high protein in the gym. No, for real, right? It's like the nitrogen is like the high protein in the gym. So now let's say someone is like very skinny and doesn't have a muscle in his body, goes to the gym. Even yourself, if you go to the gym and you drink a high protein, some sort of high protein with the you use it something prescribed with the advice of a professional, and if you drink the high protein, what happens? you will gain more muscles and you'll be growing really fast. That is because of the nitrogen. You know, nitrogen is one of the very nasty molecules in the biomolecules. You know, the nitrogen has much more effects in your body. So the one of the main thing is like you are growing fast when you have the nitrogen in your body. So it's the same for the plants, but the plants can't take any of the high proteins, right? They are different. So instead of like taking like as we take the high proteins, what does the plants take? They take the nitrates or the dissolved nitrogen type in the soil that they can absorb through their roots, you know. So that is called nitrates, right? So the nitrates in the soil is very, you know, less. If nitrogen was very like if nitrogen could find very easily, the plants would have grown really fast. You know, these plants everywhere will grow really fast. But because of the absence of nitrogen in the soil, the plants grow really slow. So when you are cultivating something, when you are doing a farm like a farm or something, if you want to grow the plants, you have to give the nitrogen fertilizers, right? You have to give the nitrogen fertilizers to grow them fast, to have a better crop, right? But if you didn't do it, if you didn't give the nitrogen to the plants, what happens? The plants won't grow, you know, the plants won't grow. But now there's an issue. The usual way how we give the nitrogen to the soil is this chemical fertilizers, right? Chemical fertilizers. So the recent government was also one of the president, he did the same thing, right? He cancelled all the nitrogen fertilizer imports, and all the farmers were like fled. So because of that, even the government was completely that was taken down by the people in the country. Yeah, go home happened, right? Yeah, so that happened because even that happened because of this. It started with something like that. So that is important. Now this is the thing. Listen. Listen. So this is the thing. The issue is when you apply the chemical fertilizers to the ground, you think the ground, like the quality of the soil is growing or losing it? When you apply the chemical fertilizers, the quality of the soil is losing. It's not growing. You know, it's completely losing, and eventually it will become a disaster. So they are calculating by 2050 there will be a soil crisis, like the economic crisis in the Sri Lanka, right? There will be soil crisis now for real. There will be limited ground to grow the plants because the soil is out of its quality, doesn't have any of the nutrients in its soil. Why? Because of the chemical, like majorly because of the chemical fertilizers, because it's not a good thing. Because when you apply the chemical fertilizers, the first thing what happens is they will remove the complete microbial flora. There will be no microorganisms in the soil. So the microorganisms are the ones which returns the soil back, you know, which returns the soil back. Because when the plants are absorbing the nutrients and when it rains and everything happens, the nutrients in the soil will fade away, you know, the nutrients in the soil will fade away. And when the nutrients are fading away, the soil becomes sand, you know, eventually the soil becomes sand. So the ones who are keeping the soil as soil itself, which means who returns the soil back when they are taking or fading the nutrients, are the microorganisms. So when you kill them completely, there is no return of the soil, because they are the ones who are degrading all the dead bodies of the animals and the plants. They are the ones who are decomposing everything and making the soil. So if you destroy the microorganisms, there will be no return of the soil when it fades away. So there is a higher and a greater risk that the scientists are calculating. By 2050 there will be soil crisis. There will be limited lands to cultivate the foods that we need for the agriculture, right? So there will be a huge risk, and if you ever be able to address that risk, You'll be the richest person in the world, you know, because the situations like that make the you know, the people, like they find the capable people in that situation as well. That is one thing. So if you are capable to find a solution for this, you know, the soil crisis which is coming in the future, because like mainly because of the chemical fertilizers, and there is one thing that you are saving the world in one way, and in another way you are becoming a rich person, because if you found it, you can earn with it, right? Definitely you can earn with it, and you'll be the richest person in the world by that time, right? So now this is the thing. So we have to anyway supply the nitrogen need of the soil, right? We have to supply anyway the nitrogen need of the soil. So there's now the what is the something you have to understand, the difference of you know, the difference of the organic fertilizers, right? The difference of organic fertilizers and the chemical fertilizers, right? So the issue of these organic fertilizers, that what the previous president didn't understand was, the problem with this organic fertilizers is it doesn't have enough nitrogen in it. Mostly the organic fertilizers contain only up to 5% or 7% of nitrogen in its fertilizer, and when it comes to the chemical fertilizers, more than 25%, you know, more than 25% of nitrogen is there. So compare the two fertilizers. Organic fertilizers have like only 7%, the chemical fertilizers have 25%. So when you apply it to the plants, when you apply the chemical fertilizers, the plant grow really fast, you know, the yield is very much. But when you apply the organic fertilizers, there will be not much of a yield. So that is affecting the farmers, ne? If there's no yield, there's no point of growing the plants, ne? Doing the business, right? So there's a huge issue. Like අපිට එහෙම කියන්න බෑ දැන් chemical fertilizers ගහන්න එපා කියලා. If you don't do that, the yield will be very low. So how do they survive, ne? Even how do the people who wants these food survive, ne? They have to have the foods, ne? There will be ඒතර ගොඩක් ගණන් වෙනවා foods අඩු වුණාන් පස්සේ, ne? ඒතර ඒවට address කරන්න ඕන. So that is the main reason that you are having this knowledge about the microorganisms and the food industry, because you will be working in this industry in the future, and you have to find the solutions for the crises and the issues that are upcoming. You know, there is one major reason. There will be so much of crises and issues coming up in the future for sure, right? So you will have to have this knowledge to address them. That is one of the reasons. So the chemical fertilizers anyway we will have to not completely remove, but we have to mitigate. We have to tail off it. Tail off means like you have to gradually remove it, not to take it off at once. If you do it at once, that is a disaster as well. That is what the previous government did, right? They removed it at once, and all the farmers and the food chain, everything corrupted. Yeah, it was like completely destroyed. Can't do it like that. So now you have to mitigate it. When you are mitigating it, when you are tail off it, you have to replace something with a better knowledge, some new sciences, and something effectively can increase the yield, but should be organic as well. So this is one of the things that you can do. But you can improve the nitrogen fixing bacteria. Now we have 78% of nitrogen in the atmosphere, right? If we improve the nitrogen fixing bacteria in the soil, maybe a genetical way, no, maybe genetical engineering or something that you'll be learning, the BST people are learning maybe in the future. So if you, BST people are supposed to learn these things, I hope, and the people who are doing agri, they're supposed to learn these things. And if you could ever improve these microorganisms that can, increase the nitrogen percentage in the soil, we don't have to take the chemical fertilizers anymore, right? If you found a way to improve the nitrogen percentage in an organic way with the use of microorganisms, that will be marvelous, right? That is saving the earth. But there could be side effects as well. We have to consider those as well. But for now, it will be a really good solution if you can improve the nitrogen fixing microorganisms in the soil to fix more nitrogen and to improve their distribution around the soil, because nitrogen fixing bacteria are not much available in the soil everywhere. So you have to improve them. So if you think of it, you can do it in the future, right? So now who are the nitrogen fixing bacteria and what do they do, right? So the nitrogen fixing bacteria are converting atmospheric nitrogen into ammonium ions, which the plants can easily absorb. The atmospheric nitrogen the plants cannot absorb, they can't absorb the nitrogen gas itself. But if you convert the nitrogen gas into ammonia, you can easily absorb. I mean, you means the plants can easily absorb. So what this nitrogen fixing bacteria does is, they take the atmospheric nitrogen to their body using an enzyme called nitrogenase. Using an enzyme called nitrogenase, they convert this nitrogen into ammonium ions, ammonium ions. So those ammonium ions the plants can easily absorb and grow, right? That is what the nitrogen fixing bacteria does, right? So come to the topic back again. Certain microorganisms convert nitrogen to ammonia, right? Convert nitrogen into ammonia using energy obtained by ATP, and nitrogenase enzyme is called biological nitrogen fixation. That is biological nitrogen fixation, and that is done by the certain type of bacteria. Atif is asking, so if there's so if we use a supplement or creatine, is there any side effect? Yeah, of any of the products that we usually use have side effects, but there is something to consider with time. The side effect takes time. If a product was released, if it has a history of 20 years back at least, you can confirm what are the side effects. But if a product doesn't have much of a history, let's say the product is very new, you can't tell about the side effects. How do you know? You have to wait until 20 years. So if the product after 20 years is still good, some good products are there, they have been there for like many years, 40 years, 50 years, they are continuing the quality. And if those things are still there in the market, you can actually find the side effects and can ask the people who used it, and it will be really fine if it is still using by other people. But if it doesn't have any of the history, you can't tell. So rather than using the new ones, newer ones, you can trust on the ones who have established their quality for several years, and better you're trusting them. And the organics are the best, Yeah. Same. You can't use the steroids as well. If you use the steroids, what happens? Your testicles are also producing sort of a steroid, that is a hormone, and your testicles will start to think, we don't have to produce this anymore, because it's already there in the blood, we don't have to produce it anymore, and your testicle will start to shut down. Now for real, the people who are taking the steroids, that will happen, right? So these are the examples, right? Azotobacter, Clostridium anaemia, right? Then the non-symbiotic anaemia species, Nostoc, Rhizobium, right, are the examples given, right? So these bacteria are the nitrogen fixing bacteria in the soil. So usually these nitrogen fixing bacteria are two types. What are the two types, you think? What are the two types of nitrogen fixing bacteria? One is, I'm asking, there are two types of nitrogen fixing bacteria. The free living and symbiotic ones, right? So there are two types of nitrogen fixing bacteria. One set of microorganisms are free living. Free livings mean they don't have any of the relationships with any of other species. They just only live on the soil. And the ones who are having the symbiotic relationships, they have a relationship with another species. Like like we discussed in the omnipresence symbiotic relationships, some of the microorganisms live with with a relationship with another species, like the Rhizobium. Rhizobium are living in the root nodes of the legume plants, right? That is a symbiotic relationship. Likewise, some are living with another species and some are living by their own self as well, right? So there are two types. So Acerobacter, free living heterotrophic aerobic bacterium. Clostridium, free living heterotrophic anaerobic bacterium. Anabaena is a symbiotic one, right? Anabaena is one of the symbiotic microorganisms. Anabaena are three types. Anabaena azulea are there, Anabaena cycadarum are there, and ඒ ඔක්කොම ඉන්නේ symbiotic relationships. මම ඒක ගොඩක් ෂෝට් කරා මේක. එච්චරම ඇවිල්ලා නැති හින්දා. හරිද? Right. So shall we highlight this Acerobacter? මෙන්න මේ free living ද නැද්ද කියන එක අපි highlight කරන් යනවා. So Acerobacter are free living. Clostridium are free living. Who else are free living? Non-symbiotic Anabaena species are free living. Then the nostoc, free living. Rhizobium is symbiotic. So there are two types of symbiotics. Who are the two types of symbiotics? One is Rhizobium, the other one is Anabaena, right? ඒ ටික highlight කරන්න separately. Symbiotic and free living කට්ටිය වෙන වෙනම highlight කරන්න. Have you people received the tutes? Arkinder, Malchi, Atif. Happy? There was a complaint. Even these people haven't received it, right? Yeah, so Malchi didn't. Yeah, so there's a complaint that you didn't receive the tutes. So by today or tomorrow, I'm going to send it by myself, and I'll personally look at it, and will surely send it by today or tomorrow for sure, and I'll be fast එවන්න පුළුවන් එකේ මම දාන්නම්. හොඳයි? Promising. Within 2-3 days, you'll be receiving your tubes. Right? Right. Next thing, come to the bacteria. Come to the bacteria. Now even you bought this to eat, right? For me? It's a gift? Huh? This is a new one? The old one, right? Right. So come to the new topic, the bacteria. Right? The bacteria. So yes. Somebody says sir, sir. Somebody says sir. How is it? ඇවිල්ලා මම පාඩම් කරලා තිබ්බට කමන්නේ පාඩම් කරලා තිබ්බට කමන්නේ What is ATP? Now Malchi is asking what is ATP? That is adenosine triphosphate. There is a sort of a chemical that stores energy in a chemical format. You know, that is a sort of a chemical that stores energy, ATP. Energy storing chemical, adenosine triphosphate. Oh, ATP adunam cramps, you know. That is true, right? So come to the new topic, bacteria, right? Bacteria, and this is really important. And there these features, you'll have to study everything You know, you'll have to remember everything of these features, right? So now come to the bacteria, right? So right. Right, come to the bacteria topic. So these are the main characteristics. First thing is they are prokaryotic cellular organization, right? So what is the cellular organization of the bacteria? Highlight it. They are prokaryotic. Prokaryotic means they don't have an organized nucleus, right? The prokaryotic means they don't have a organized nucleus, right? That is what it simply means. What is the meaning of prokaryotes? Prokaryotic means, bro in the back row, you say what is prokaryotes? What is the meaning of prokaryotes? It's not there in the tube. What? Cellular what? Cellular your ass. You listen. Right? You first thing is you listen when I'm speaking. That is the first thing. Right. So, what is prokaryotes? Prokaryotic means they don't have an organized nucleus. They don't have an organized nucleus, right? They don't have a membranous organelles. That is what it simply means. Their nucleus isn't covered with the membrane. They are all floating in the, the plasma of the cell, like randomly. And when it comes to the eukaryotes, they have a membranous nucleus. They have a membranous nucleus that is covered and organized. Right. So the first thing about the prokaryotic means is like they don't have a organized nucleus, right? Second thing is they are found in everywhere in the biosphere. That is called the omnipresence. Bacteria are everywhere. Like wheresoever you go in the biosphere, they will be there. There's nowhere that is absent, right? They are the most abundant living organisms on earth. What does it mean? They are the most abundant. They are the most available organisms. They are the, mostly available organism, not the microorganisms, the organisms, the mostly available organisms in the earth are the, the bacteria, right? There's a huge number. And their size varies from 0.25 micrometers to 5 micrometers, and this is really important. This part is really important. Highlight it as well. Their size is between 0.25 micrometers and 5 micrometers, and above. So above the size of 5 micrometers, they are known as the eukaryotes, right? So up to this level, these are prokaryotes. This is the size of a prokaryote. And above this size level, they are all eukaryotes, right? They are all eukaryotes, right? Next one, shows a higher reproduction and growth rate. Yeah, they show a higher reproduction and growth rate as we discussed earlier. Bacteria set generation time. So they have a rapid growth rate, right? That is one characteristic. They are either unicellular or colony forming. Unicellular means they are only single cell. They have only a single cell as their whole body. The whole body is a single cell. Very small people, not the people, the microorganisms, right? Their whole body is a simple cell. And they are not multicellular as well. Multicellular means they don't have number of cells in their body. They only have one. And another way of their living is a colony. Colony means number of cells living together as a cluster, you know. Yeah, yeah, huh? අන්න ජනපදයක් තමයි. ජනපදයක් කියලා කියන්නේ ඒ වගේ තමයි. හරි colonies somewhat ජනපදේ තමයි. නේ colony කියන්නේ ජනපදේ තමයි. ඔව් සිංහලෙන් ජනපදේ කියන්නේ colony, right? කියන්නේ colony එකක් විදිහට ඉන්නේ. අපි අර කියන colony එකක් කියලා. It's the same, right? Colony, right? So they either live as single cells. Now this unicellular means single cells. අපි ඒකත් ලියාගමු. Unicellular කියන්නේ මොකද්ද? Single cell. Either they are unicellular or colony forming. Colony means a cluster of cells living together. You know, cluster of cells living together. The next thing, show a higher diversity of respiration and nutrition. Show a higher diversity of respiration and nutrition means they have 4 of the respiratory types and 4 of the nutritional types. We discussed those. What are the 4 of the nutritional types? Autotrophs, then the chemoautotrophs, photoheterotrophs, chemoheterotrophs. And what are the 4 of the respiratory types? The aerobics, facultative anaerobics, microaerophilic and obligate anaerobics. ඒ තියෙන types ඔක්කොම පෙන්නනවා කවුද? Bacteria. They show all the types. Show higher diversity of respiration and nutrition. That is true. And this is also true. Yeah. Most of the bacteria are chemoheterotrophs. මේකත් important. කියන්නේ bacterias වල අර types හතරම පෙන්නුවාට most of them, the majority of them are chemoheterotrophs. That is nutritional types. And when it comes to the respiratory types, what is the major respiratory type that the bacteria are showing? Respiratory types. Hetero are the digestive types, nutritional types. Respiratory types, aerobic. If it wasn't aerobic, you can't live on earth. Why? If it wasn't aerobic, because in the aerobic respiration they only produce carbon dioxide and water, which is not much affecting to ourselves. I mean, like generally they don't affect us. But when it comes to other things, fermentation and the lactic acid production, methane production, respiration and having, if everything was fermenting around us, can we survive? We can't. If everywhere was methane, we can't survive. If everywhere was like lactic acid, we can't survive. So the majority of the bacteria are doing aerobic respiration, and because of that we can peacefully live. Otherwise any of other type of bacteria increased, we can't survive, because they will be producing much more harsh things to live. Yeah, lactic acid, methane, alcohol, ethanol. Except for the ethanol, all the other things are hard to live. Even if the alcohol level is high, you can't survive in that sort of condition. You will die for sure, right? So most of the bacteria are chemoheterotrophs, right? The next one, they have the ability to fix atmospheric nitrogen, and some of the bacteria can produce toxins, right? We already know that they can fix the nitrogen, and some of the bacteria produce toxins. Some of the bacteria produce toxins, and these toxins are 2 types. One is a endotoxin, the other one is a exotoxin. So it's a simple thing. Endo means inside, exo means outside. So the endotoxins are the toxins, the toxins, right, which could be poisonous for the whoever that it takes to their body. So endotoxins are produced inside their cells. So if you take the whole microorganism into your body, it will get toxic, right? And the exotoxins are, they produce it and they release it to the surrounding, and if you take anything in that surrounding into your body, it will get toxic to you, right? So those are endotoxins and exotoxins. So usually they produce them for their own protection, because the other animals are coming to eat them. කන්න හදනවා කියලා කියන්නේ එහෙම වෙනවා. And to protect themselves from the others, they produce these toxins. So they don't eat them. Yeah? Right. So exotoxins and endotoxins are the two type of the toxins they produce. And the reproduction is binary fission. So what is binary fission? What is binary fission? Huh? Now let's say tiny is now he's all of a sudden is vibrating, and it makes a fission in the middle and divides into two and becomes too tiny. That is binary fission. Huh? Like then අපි හිතමු මේව vibrate වෙන ගමන් මැද්දෙන් fission එකක් හැදෙනවා. One of the fission that may be a wall or something, a sort of a wall is produced in the middle, right? And it divides into two and produce two equal tinies. That is binary fission. So that usually happens in the bacteria, sort of microorganisms. So what is binary fission? Yeah, listen, I'll be asking much more questions from you from now on. You tell what is binary fission? What happens? It makes a fission in the middle and breaks into two and makes two equal products of the same bacteria. That is binary fission, and that is how they reproduce. How they Gadavilla don't only the primary living show the binary fission. You don't call it that is stigma. That's a different case. That is stigma, and even එහෙම ගැඩවිලා දෙකට කැල්ලා දෙන්නෙක් හැදෙන්නේ මැරෙනවා. Don't do that. Now home, making it home, making it feel like home. That's why I call the homie. Right? Right. Reproduction is binary fission, right? Binary fission again you can divide right? Some of them are causing disease. Some of them are causing diseases. Those are called the pathogens, right? Some are pathogenic. We call it pathogens, right? The microorganisms who cause the disease, we call them pathogens, right? Pathogenic to plants and animals. And all of the nutritional types are also there. All nutritional types are present, right? All nutritional types are present, right? All nutritional types are present. All the nutritional types are present. Right, come to this one. Structure of the bacteria cell, right? Hurry. So we'll have to draw a structure. We'll draw a structure and we'll have to label it. Structure ka draw kala label karamu. So it's easy to understand. So these are the structure of a bacteria cell. Listen, this is important. The bacteria has a cell wall, right? Bacteria has a cell wall, and that is made out of a sort of a chemical called this what? Peptidoglycan. Peptidoglycan. This was there in many of the questions. Peptidoglycan. So what is the bacteria cell wall made of? Bro, the specs. The specs boy. Your name, I can remember your name. Huh? Hansamal. Hansamal. Hansamal, tell. what is the bacteria cell wall made of? Petty. That is peptidoglycan. Peptidoglycan, right? Now this will be there in your questions, huh? This will be there in your future questions, huh? Peptidoglycan. Peptidoglycan, right? The name is peptidoglycan. What is the chemical compound that the bacteria cell wall is made of? Itra answers කීපයක් තියෙනවා. Cutin, chitin, peptidoglycan. Peptidoglycan, it's one name. Peptidoglycan is one name, right? Second, some of the bacteria can secrete a lipid capsule out of the cell wall immediate inside of the cell wall. Yeah, so there should be a comma here. Some of the bacteria can secrete a lipid capsule out of the cell wall, right? Some of the bacteria can produce a lipid capsule out of its cell wall. Then that is for their protection, you know. In some of the bacteria there is this lipid capsule around them, you know, that when that lipid capsule is there, none of the chemicals, heat or sometimes the radiation can't kill them. Some of the radiation can't kill them, so they, like, when there's a heart condition, they tend to produce these outer capsules of lipid to survive, you know. That is the lipid capsule they are producing outward the cell wall. Some of the bacteria can secrete a lipid capsule out of the cell wall. And immediate inside of the cell wall, plasma membrane, the cytoplasm are present, right? Plasma membrane and the cytoplasm are present. Cytoplasm are present, right? Cell wall එක ඇතුලේ තියෙන්නේ immediate inside the cell wall තියෙන්නේ මොකද්ද? Plasma membrane and the cytoplasm are present. Cytoplasm ඒ කියන්නේ cell wall එක තියෙනවා, immediate inside the cell wall, plasma membrane, then the cytoplasm. එළියෙම තියෙනවා එතකොට මොකද්ද? cell wall එකට එළියෙන් තියෙනවා capsule එක. So the capsule, cell wall, plasma membrane and cytoplasm. හරිද? තමයි layers ටික තියෙන්නේ. Some of the bacteria contain a flagellum or a microphyllae out of the cell wall. හරි. සමහර අයගේ තියෙනවා flagellum එකක්. What is a flagellum? It's like a tail, නේ? It's like a tail. They use it for their locomotion, right? We call it the locomotion. The movement, they use it for the movements. We call it the locomotion. Yeah, likewise. Like to swim. And some of the bacteria, yeah, their genetic material is made of DNA and small circular DNA called what? Plasmids. These are important, and because we will be discussing this back again in another part of this lesson. Plasmids. Now, like, apart from these DNA molecules, there are plasmids as well. So what are the plasmids? Plasmids are the small DNA circles, you know, like apart from the major big DNA, there are small plasmids are there. ඒක හරියට නිකන් අර ලොකු sugar cane මේ ලොකු crystal එකක් එක්ක චූටි crystals පොඩ්ඩක් කීපයක් තියෙනවා වගේ. single strand DNA RNA නේද? ඒ කියන්නේ සාමාන්‍යයෙන් double strand තමයි DNA තියෙන්නේ. ඔව්. ඒ කියන්නේ ඒගොල්ලන්ගේ අර මේ eukaryotes ලගේ තියෙන DNA තරම් complex නෑ. ඒක නිකන් තියෙන දිගහැරලා තියෙන. දැන් ඒ ටිකේ ගොඩාක්ම details ඕනේ නෑ අතර. මොකද දැන් questions refer කරාන් පස්සේ ගොඩාක් එහෙම details අහලා නෑ. තේරුම් ගන්න. හරි. එතකොට මේගොල්ලන්ගේ the genetic material is made up of DNA and small circular DNA called plasmids. It's a must to remember the plasmids. Plasmid කියන්නේ small circular DNA එකක්. චූටි රවුමක්. අර මේ එකක් වගේ මොනවාද? ඩෝනට් එකක් වගේ. චූටි ඩෝනට් එකක් වගේ රවුමක් තමයි තියෙන්නේ plasmid කියන්නේ. හරි. Plasma membrane is made of phospholipid bilayer and the folding on the මේක highlight කරලා ඉන්න. plasma membrane is made of phospholipid bilayer. හරිද? ඒක හැදෙන්නේ phospholipid bilayer එකකින්. ඒ කියන්නේ දැන් මේ තියෙන plasma membrane එක නම් මේ කැලල අපි ලොකු කරලා ගත්තොත් ඒක හැදලා තියෙන්නේ මෙන්න මෙහෙම bilayer එකකින්. හරිද? ඔන්නෝම bilayer එකකින් හැදෙන්නේ. layers දෙකකින් හැදෙන්නේ. මේ තියෙන phospholipid bilayer එක. හරිද? මේ තියෙන මේ එක layer එකක්, පල්ලෙහා තියෙන එක layer එකක්, උඩ තියෙන්නේ එක layer එකක්. මේ තියෙන පොඩි කැලලක් phospholipid එකක් කියලා අපි කියනවා. හරිද? ඔකට අපි කියන phospholipid bilayer එකක් කියලා. ඔහොම දෙකකින් තමයි හැදෙන්නේ. කැලලි දෙකක් තමයි තියෙන්නේ. phospholipid bilayer එක බෑන්ඩ් එකක් දැම්මා වගේ තියෙනවා. right? ෆුල් එක හැදෙන්නේ. ඒ කියන්නේ මේක කම්ප්ලීට් වෙනවා සර්කල් එක වටේට. කම්ප්ලීට් සර්කල් එක එනවා. හරිද? right. And the folding on the plasma membrane called mesosome has the ability to respirate. හරිද? plasma membrane එකේ තියෙනවා folding එකක්. ඒ කියන්නේ දැන් මෙයා plasma membrane එකනේ. මම මේක ආයෙ අඳින්න නැත්නම් මේක මැකෙනවා සම්පූර්ණයෙන්. මෙන්න මේ plasma membrane එකේ මෙන්න මෙහෙම ඇතුළට ආපු පොඩි folding එකක් වගේ තියෙනවා. හරිද? මේ folding එකට කියන මොකක් කියලාද? mesosome කියලා. මේ folding එක ඇතුලේ තමයි ඒගොල්ලන්ගේ respiration කියන එක වෙන්නේ. ඒ කියන්නේ සාමාන්‍යයෙන් cellular respiration වෙන්නේ කොහෙද? What is the organelle that does the cellular respiration? What is the organelle? Mitochondria, isn't it? Mitochondria does the cellular respiration. But in eukaryotes, sorry, in prokaryotes, they don't have membranous organelles. So they don't have any of the mitochondria. So where do they do the respiration? They do the respiration in the plasma membrane inside of a pocket of made of like it's like a perk like came out of the plasma membrane, and that is called the mesosome. So the respiration of bacteria occurs at mesosome, not the mitochondria. Right? The respiration of the bacteria occurs at mesosome, not the mitochondria. Right? Right. Mesosome has the folding of the plasma membrane called mesosome has the ability to respirate. Right? Has the ability to respirate. Right. As the food serve, glycogen and volutin are present. Right? මේක තමයි important න මේගොල්ලන්ගේ food serve එක මොකද්ද? Glycogen. Apart from glycogen, volutin is also present. ගොඩක් වෙලාවට අහන්නේ ඔයගේ glycogen එක තමයි. හරි. ඒගොල්ලන්ගේ food serve එක විදිහට තියෙන්නේ මොනවාද? Glycogen. Glycogen is the food serve, right? And the other thing, 70S ribosome are present. So there are two types of ribosomes. One is 70S, the other one is 80S. Now the 80S is larger, the bigger in size. 70S is small in size, right? So the 70S is only present in the prokaryotic cells. The 70S is only present in prokaryotic cells, which are the bacteria and archaebacteria. Yeah, and this 80S is present in eukaryotic cells, right? So there are two type of fungi are the eukaryotes. Oh, all the eukaryotics have this 80S ribosome, right? So if it is 70S ribosome, that is present in the prokaryotic cells, which are the bacteria and archaebacteria. If it is 80S, that is present in the eukaryotic cells, right? ඒවා තියෙන්නේ eukaryotic cells වල විතරයි. ඒකත් මතක තියාගන්න ඕනේ. 70S තමයි මේගොල්ලන්ගේ තියෙන්නේ. 70S තියෙන්නේ prokaryotes ලා හින්දා. 70S ribosomes. Bacteria type called mycoplasma doesn't contain a cell wall, right? Mycoplasma කියන කට්ටියගේ cell wall එකක් නැහැ. මේ question එකක් තිබ්බා MCQ එකක් විදියට. This was also there. Mycoplasma කියන කට්ටියගේ cell wall එකක් නැහැ. Usually they have a cell wall, but it is very thin, and because it's very thin, they neglect it. You know, that is what they do. ඒක ගොඩාක් පොඩි. So they neglect it. Sort of thing. Right. Come to the next topic. There are two types of bacteria depending on the gram staining, and this is important, right? Why do we do the gram staining? Why do we have to do the staining? Did you ever like have heard the staining of the microorganisms? No. They stain the microorganisms. Even in the prepared specimens that we observed in the first week. Can you remember? So that prepared specimens were pink in color, green in color. You saw it, right? They were like prepared specimens of microorganisms in my microscope. Were you absent? We'll do a practical, right? So adanimi we'll do a practical. These microorganisms we observed have color. They don't naturally have colors. So what do we do? We add colors so they are more visible to the microscope when you are observing them, right? So that's what we do. That is called staining. It's called staining. We add the colors to them, right? And one of the not like that, not like the literature way. We add the literal colors, the original actual colors to the microorganism, so it is easy to observe. So that is called staining. So one of the way of staining is gram staining. Gram staining is one of the stains. So there are several type of different type of stains. So gram staining is one of the stains type, right? So now this gram stain, this is the thing. In the gram stain, when you apply the gram staining, the gram stain attach with the cell wall of the bacteria. So if there is more peptidoglycan, or if the cell wall is more thick, more of the stain will attach with the bacteria, and the bacteria with a greater cell wall, a thick cell wall, will appear very colorful, and the bacteria with low percentage of this cell wall or the peptidoglycan will appear light in color. So you can identify two different type of bacteria depending on the thickness of the cell wall, which we call the gram positive and gram negative, right? So gram positive means the cell wall has more than 10% of peptidoglycan in it, which means the cell wall is very thick. So when we gram stain it, they will appear in purple, because the stain is purple in color, so they absorb much more colorants into it, so they appear in purple, gram positive ones, because they have a thick cell wall. And if you stain the gram negative bacteria, which have a very small cell wall and less amount of peptidoglycan, as in theory we say less than 10%, right? If there's less than 10% of peptidoglycan in their cell wall, they appear in not purple, they appear in pink. That is when you light the purple like appears like pink. That is why it calls like pink. So the gram negatives appear in pink color, gram positive appears in purple color. That is how you identify two type of bacteria, and the gram positive are more pathogenic and hard to destroy. There is a difference, right? Right. So there are two type of bacteria depending on the gram staining. One is gram positive bacteria, the other one is gram negative bacteria, right? Positive and negative. And the gram positive bacteria is consists of peptidoglycan is more than 10%, right? Peptidoglycan is more than 10%. They are called the gram positive bacteria. And in the gram staining, the stain highly bound with the peptidoglycan and shows a violet color or a purple color. Gram positive ayage pellane violet natham purple color එක. When observed in the microscope. Come to the gram negatives, right? These bacteria contains less than 10%, right? These bacteria contain less than 10%. of peptidoglycan in their cell wall. They show a purple or pink color when observed to a microscope after the gram staining. They usually show a pink color, right? If it is pink, that's a gram negative bacteria, right? It's a gram negative bacteria. Right. So close your tube now for a second. For a few minutes, close the tubes. People, close the tubes. For a few minutes, close the tube. I want you to remember. Close the tube. Yeah, close the tube. Yeah. Even the online people, I think they don't have the tube kit, but if you have it, please close it. Huh? Yeah. I'm sending it by today or tomorrow. Right? Close the tubes. Right. So now I want you to remember the complete structure of a bacteria. I'm going to draw it by myself, and you follow it, you keep it in your mind, and you label it by yourself. Yeah, draw and label it by yourself. Here it comes. Yeah. Huh? Yes. That one is. Close it. Oh. Right. So now this is a bacteria cell. It has few layers. 1 layer, 2 layer, 3 layer. It's a big fat bacteria. Right? So there's 3 layers, and this is the main DNA molecule, right? And apart from that, a small circular DNA is there. What do you call this? What do you call this DNA? The donut shape, huh? Plasmid, right? You call this a plasmid. You have to remember those, huh? Then apart from these plasmids, what do you think these black dots are? The black dots are there. These are what? Yeah, which ribosomes? These are the 70S ribosomes, right? So apart from these 70S ribosomes, there's another thing in the plasma membrane. As we discussed, there's a small folding of the membrane, right? There's a small folding of the membrane here. මෙන්න මෙතන තියෙන පොඩි folding එක. What do you call this folding? Huh? What? No. The bilayer itself is the plasma membrane, but in the plasma membrane there's a folding. Huh? No. Mesosome. Can you remember? Mesosome. Mesosome is the respiratory body of the bacteria. This is the mesosome, right? So most outer layer is what? Most outer layer is the capsid or the lipid capsule, and inside the lipid capsule there's cell wall. Inside the cell wall there's plasma membrane. Inside the plasma membrane there's cytoplasm. In the cytoplasm there is a main DNA, right? And apart from the main DNA there's a plasmid. And what are these? 70S ribosomes, and this is mesosome. So together how much? 1, 2, 3, 1, 2, 3, 4, 5, 6. Yeah, some sort of 9 are there. So shall we label it now? Now this is what? The capsule, right? This is the capsule. And is green. This is the capsule. ඊට පස්සේ that is the most outer layer. මේක කවුද? This is the cell wall and made of peptidoglycan. And ඒකත් ලියන්න හොඳ. This is made of peptidoglycan. Right. And inside of the cell wall there's plasma membrane. Plasma membrane is there. Plasma membrane. And inside the plasma membrane there's cytoplasm. Cytoplasm. And this is main DNA molecule. Main DNA molecule. And this one is a plasmid. There can be more than one plasmid. But there is only one main DNA molecule. Can be more plasmids. And this is 70S ribosome. So how many are there? This is what? This is mesosome. Mesosome. So all together 1, 2, 3, 4, 5, 6, 7, 8, 9 are there. Right? So 9 are there. So now take 1 or 2 minutes and remember the whole picture and draw it by yourself without watching the board or without watching the tube. Anam, take a piece of paper. The pillai is also there. Yes, yes. There's more to draw. Listen. So there's a pillai. Come on. Tube video center come on. Tube video center come on. There's a flagellum. This is called what? Flagellum. This is a flagellum. And apart from the flagellums, these pili are also there. These are the pili. यार රැවුල වගේ තියෙන්නේ රැවුල වගේ තියෙන්නේ මොනවාද රැවුල වගේ තියෙන්නේ පිලයි. This you call the pili. Right, this you call the pili. රැවුල වගේ තියෙන්නේ පිලයි. So altogether 11 are there, ne? Yeah, 11 are there. 11 are there. So you have to know to draw and label all the 11 parts. So usually the structure will come, you will have to label. How the structure you will have to label. But it's a good thing if you can draw the structure because it's easy to remember if you draw it. How is it? Now you finish? Shall I remove it? අඳින්න නෙමෙයි කිව්වේ බලා ගන්න. බලලා මතකෙන් අඳින්න. What the hell, why do you want to like draw මේක චිත්‍ර එකක් කියලා? චිත්‍ර තරගයක් නෙමෙයි. Just have to මේක දිහා බලන්න, memorize it. අතන ඉවරයි. මතකයිද? මේක අයින් කරනවා. දැන් ඔගේ බලාගෙන යනවා you remove it. There's no point of doing that. Remove it. Good. Throw it away. Yeah. Do it. Method cytoplasm. Finish? can't draw it. හරිනේ බලන්න බෑ. හා මතකයිනේ? හා ඉවර කරන්න බලන්න. Rajalam. What? Pilai. Pilai. Just pilai. Right? Finish? Yeah. There's only 11. Now people online, take a piece of paper, just don't draw anything now. Try to remember it, the whole thing. I think this is disturbing, right? Right? Enough time. Draw and label quickly. බලාගෙන ඉන්න නෙමෙයි දුන්නේ. පොටේ පිටිපස්සේ කමන්නේ. Finish? Huh?
डायग्राम में संपूर्ण रहेंगे तो नाम कराएंगे
Looks like an alien. Cool label name. Okay.
मैं मेकर सेकंड ट्राई
Ah, he's doing his own job. You do your.
120
Ah, bro, use a pen. That's a garbage paper. Use a pen. You don't even have a pen. මේ කරන්ට් එක. මේ පිග්මන්ට් එක දාලා තියෙන්නේ.
දාලානේ තියෙන්නේ. ඒකට මේකේ ඇත්තටම පාට නෙමෙයි.
ඇත්තට මේකල කලර් එකක් නේ.
දැන් නෑ මම සර් පින්ක් පාටයි පර්පල් පාටයි
ආ නෑ නෑ මේ මේකල පැරමීසියම් බැක්ටීරියා නෙමෙයි. බැක්ටීරියා හොම් බලන්න බෑ. හොම් පේන්නේ.
මොකද බැක්ටීරියා වල පාට පාට.
පැරමීසියම් එක බැක්ටීරියා වලට ගොඩක් ලොකුයි.
බැක්ටීරියා පොඩි වැඩි.
ගොඩක් පොඩි. ඒකට පව එක වැඩි කරලා බලන්න ඕන තව.
පාට පේන.
කලර් කළා තිබ්බ පේන.
सर එතකොට ඒක කලර් කරලා තියෙන නැත්නම් ඔය හැටි
මේ කලර් කළා තියෙන්නේ. Is it pen? Now somebody give this guy. No. Give him a pen. Not that one. Yeah. Girls, give this guy in the back that bigger a pen. Give a pen. Pen ගන්න විදියක් නෑ. pen. Right, finish? Yeah. කීයක් තිබ්බද? 10ක්? 10ක්. කොයි එකද? Flagellum there, capsule is there, villi is there, cell wall. Cytoplasm එක තියෙනවද? ආ තියෙනවා cytoplasm.
මේ මයිටොසිස්
ආ mesosome.
සර් කොහොමද
මේ ටේබල් එක ටියුට් එක අලව ගන්න. කපලා අලව ගන්න. Right. So what are the parts? හා මේ ටික තියෙනවා නම් දාගන්න හරි 10ක් තියෙන්න ඕන. 10ක් right 10ක් තියෙන්න ඕන. තියෙනවා හරි 10ක් තියෙනවා හොඳයි. It's good thing. Right. හරිද බලන්න මෙන්න මේ පාර්ට්ස් එක තියෙනවද කියලා. හරිද? මේකේ අර මේ එක ලේබල් කරලා අපි ඒකින්ද ඒකත් මෙතන ඇදගමු. ඒතර මේකේ තියෙනවා මොකද්ද? ෆ්ලැජලම් එක තියෙනවා. ඊට පස්සේ මේ තියෙන මේන් DNA එක, මේ ෆ්ලැජලම් එක මේක හොඳේ. මේ දෙක මාරු වෙලාද කොහෙද තියෙන්නේ? මේ මේන් DNA තමයි මේ තියෙන්නේ. ලොකු එක තමයි මේන් DNA එක. හරිද? ෆ්ලැජලම් එක තමයි මෙන්න මේක. හරිද? මේ තියෙන්නේ ෆ්ලැජලම් එක, මේ තියෙන්නේ මේන් DNA එක හදාගන්න. ඊට පස්සේ එළියම තියෙනවා මොකද්ද? ලිපිඩ් කැප්සූල් එක, ඊට පස්සේ සෙල් වෝල් එක, ඊට පස්සේ ප්ලාස්මා මෙම්බ්‍රේන් එක, ඊට පස්සේ සයිටෝප්ලාසම් එක. මේ තියෙන සයිටෝප්ලාසම් එක. ඊට පස්සේ 70s රයිබෝසෝම් තියෙනවා, ප්ලාස්මිඩ් එක තියෙනවා, පිලයි තියෙනවා. අපි මෙන්න මේක ඇඳගමු. මොකද්ද? මේකට අපි කියන මොකක් කියලාද? mesosome. mesosome මේක ලේබල් කරගන්න mesosome. මෙන්න මේකත් ලේබල් කරගමු. මේක ලේබල් කරගන්න reserved food මොනවාද? glycogen. Reserved food. මේක මොනවාද? Reserved food. Reserved food. Reserved food. අපි කියනවා reserved food එක මොනවද තියෙන්නේ? Glycogen. Hi, reserved food එක glycogen. Right. So is there anything missing in the diagram? No, right? Everything is there in the diagram, right? Right. So the next week we will discuss the how different figurative forms of bacteria and nitrogen cycle mainly. Right, we'll discuss the nitrogen cycle mainly. This is the most important part. We will be discussing this the next week, and most probably we will be finishing this part the next week. Next week අපිට මේ part එක ඉවර කරගන්න පුළුවන්. හැබැයි අර අරගොල්ල තියෙනවා ප්‍රොටෝසෝවා පොඩි පොඩි ටිකක් තියෙනවා ඒක. ටිකක් තියෙනවා. Right. People online, then we see you. Like, put a text. Yeah, late. Match එක ගන්න ගියා. මොකද උනේ දින්න?
අද වාහනේ වාහනේ නේ මේ YouTube ඔය හරිද?
මට